theory, I am live. So I'm waiting. First of all, are you all able to hear me? I've never done a live stream before, so this one will be a little bit messy. I'm not seeing anybody in the live stream yet. If anybody's here, can you say something? No viewers so far, but it looks like everything is working. So I'll go ahead and introduce myself and see if anybody joins us. So I am Dr. Josh Banta. I am a professor of biology at a university here where I live in East Texas. My research areas are evolutionary biology with an emphasis on rare species, conservation biology. I do evolutionary and population genetics, and I specialize in computational methods. And that's how my YouTube channel came about. I make these videos primarily for the courses that I teach, primarily for the graduate courses that I teach. And I also upload them onto YouTube. And I've made quite a lot of them in the last couple of years during the pandemic because I've been teaching some of my courses in an online format. And so I've been making a lot of tutorials about how to perform the laboratory exercises and providing that to my students, and then also providing it to you here on YouTube. Looks like I've got a viewer. That's great. Hopefully we'll get more. This channel, if you're watching, you probably know, is about many different topics in conservation biology, evolutionary biology, ecology, and evolutionary genetics. So I do a lot of videos about geographic information systems, also known as GIS by its acronym. I do a lot of videos about landscape ecology more generally, especially videos about species distribution modeling. Some of my most popular videos are on species distribution modeling. You'll also see videos on this channel about population genetics, phylogenetics. I have some videos that provide background into these methods and into these approaches, into these disciplines. But most of my videos focus on actually getting the job done. Here's how to do what you're trying to do. And I make videos that typically the troubleshooting to get it done isn't provided anywhere else. For most of my videos, almost all of them, except for the really early ones, I use the Ubuntu Linux operating system, which is the one that you see on your screen. Oh, thank you, Dan. Very nice to meet you. Thank you for joining in. The reason that I use Ubuntu Linux, well, there's a number of reasons. So I don't assume a high level of really geeky computer knowledge for my tutorial videos. Oh, hi, Andres. Very nice to meet you as well. Oh, greetings from Guatemala. Hello. Greetings from East Texas. So where was I? I think I was talking about how I use the Ubuntu Linux operating system and the fact that I don't assume that my audience members, whether it's you on YouTube or whether it's the students in my graduate courses where I primarily make these videos, have a really sophisticated understanding of computers, a really in-depth understanding of the Linux operating system or even the Windows or the Mac operating system. I just assume a functional level of knowledge. And so I design my videos so that you can go ahead and use them and give you the steps and sometimes even the scripts that you need to use them to do the most common things that you're likely to want to be doing. The reason that I use Ubuntu Linux is because that way I can set up virtual machines, and I can set up those virtual machines at no cost. A virtual machine is a computer within a computer. It's kind of like the movie The Matrix, where all of the people are living in that world that's actually nested within another world. The point is that in Ubuntu Linux operating system, it's very easy for me to create a self-contained computer system that has all of the software that I'm demoing, and it has all of that software pre-installed and pre-configured. And it means that everyone who is following my tutorials, who's using that pre-configured virtual machine, will be using the same machine that I'm using in my tutorial. So this will cut down on problems where a particular computer the software is not installed right, so the analysis won't run. It really helps with the troubleshooting. It creates a uniform environment. If you use my Ubuntu Linux machine, you're using the same machine that I use. Yes, I agree, Linux is great. I have a lot of fun playing with it. I spend way too much time playing with it. It's really great if you like to get into the weeds and really understand the guts of your computer. But it has a sophisticated enough graphical user interface that you don't have to be geeky to use Ubuntu Linux. It looks a lot like a Windows or Mac user interface. So I like that too. Anyone can use it in a user-friendly way, and can even use these complicated computer programs, generally speaking, in a user-friendly way. So there's also, for population genetics and for GIS, and for landscape ecology, oftentimes the programs are actually written for Linux. And maybe there'll be a version for Windows, and maybe there'll be a version for Mac, kinda, but oftentimes the software is primarily written for Linux. And therefore, it's more native to run these software programs that I demo within Ubuntu Linux. So that's another reason. Also, there are some oldies but goodies. For instance, there's the program Harlequin, or another program called Structure. And these are old programs that they work to varying degrees on modern Windows computers. They don't work on Mac computers. Structure used to work on Mac computers, but it doesn't anymore. Harlequin has always, well, the graphical user interface version of Harlequin has always been Windows only. You can kind of sort of use Harlequin on a Mac, but you still have to go back to a Windows machine. But anyway, actually, there's a way that you can run that old Windows program within Linux. So I'm able to take some of these oldies but goodies and also include them within this virtual machine environment, along with all of the other software. That said, most of the programs that I demonstrate on my channel, and most of you probably know this, but some of you may not know this, can be run on your normal computer without using VirtualBox. Andres, I'll just interrupt my stream of thought here, because this live chat is rather a stream of consciousness anyway. Is asking if I've ever worked with phylogenomics data, and I have. I have some tutorials about it. My main expertise is with reduced representation sequencing data, specifically with RADseq and RADseq-like approaches. So they're very large data files, much larger than you could use in a program like PhyML. And so for that, I have used a program called Beast. I've used another one called RaxML, which is a scaled-up version of doing maximum likelihood-based phylogenies and beasts. is a scaled up version of Mr. Bayes. I'm simplifying here, for doing coalescence based. So the first one is for concatenation based phylogenetic methods. That would be your RAXML, and then for your coalescence based phylogenetic methods, which are typically done in a Bayesian framework, I'd use a program like Beast. And there's an add-on for that called Snap, which goes on top of Beast, which lets you work with, rather than whole genome data, with reduced representation sequencing data. So I do have experience with phylogenomics. I've never tried Rev Bayes. I think, yeah, so Rev Bayes, as I understand it, does the same kind of thing as Beast. And please correct me if I'm wrong. I know that there's sort of more general purpose Bayesian computer, statistical analysis programs, but I think Rev Bayes is another flavor of doing what I do with the program Beast. Anyway, we got sidetracked there, but in a good way. I'm happy to have the interaction. I want to emphasize again that many of the programs, and I can mention some of them here in the live stream, you don't need to use VirtualBox, but it means that you'll have to install those programs yourself on your own computer. So some examples of those would be RStudio. I have an increasing number of tutorials on here using R, using a more user-friendly version of R called RStudio, which is a everything software package for doing statistical analyses, for making graphical output, for doing phylogenomics, for doing GIS. Actually, a lot of the stuff that I demo, stuff that I demo using Harlequin or using Structure or using, if it's landscape ecology, using Grass GIS, those things have versions of those programs, of those routines that you can run within RStudio. And RStudio can be installed on your Windows machine or on your Mac machine. So if you're watching one of my tutorials that uses RStudio, you can actually just go ahead but there's going to be two things there. You're going to need to download R and install it on your computer. And then on top of it, you're going to download and install this other layer that makes it more user-friendly called RStudio. And there's lots of YouTube videos about how to download and install R, and then to download and install RStudio for Windows or Mac. So just keep that in mind, that even when you're seeing me demo those within my virtual machine environment, that you can still, without the extra hassles of having to set up this virtual machine, or I don't know, set up a Linux machine separately or whatever, you don't actually have to do all of that. Harlequin, you can just install Harlequin on your Windows machine. I think it still runs on the current generation of Windows machines, but eventually it's going to stop working. It's an old program. But that's another example of one where, even though you see me demo it on my virtual machine, that you can just go ahead and install Harlequin. Okay, great, yeah, so it still works. Awesome. Let's see here. Now, I mentioned sort of on the side that not only do I demo stuff within Ubuntu Linux, but I have everything set up in this virtual machine. You probably already know, because I say it in all of my tutorial videos, that I have the Ubuntu virtual machine pre-configured with the software that I talked to you about in that tutorial, already available for you to download. It's a very large download, so it can take several hours, but then you can actually download the exact machine that I'm working with, and using this program called VirtualBox, you can use that virtual machine within your Mac computer. So even though you're sitting in front of your Mac computer, you'll have a little window open within your Mac computer where you can run that virtual machine that's identical to the one that I'm running in the tutorial. Now, there's been an important change here, which some of you may know about, which is the new chip architecture being used by Mac computers. And that is the M1 and now the next generation, the M2 chip architecture, which is an ARM-based architecture. This is all to say that you can no longer run my virtual machine. You can no longer run VirtualBox, which runs my little virtual machine if you've got a Mac computer, unless you have an old Mac computer. So I'm running a 2-year-old Mac computer here that uses the previous architecture, I guess it's called the X64 architecture. The older style Mac chip architecture will run VirtualBox. So if you're running a Windows machine, and I mean a normal Windows machine, I need to put a caveat in there. If you've got some kind of a fancy Windows machine with an ARM chip architecture, and you probably don't. If you understand what I'm talking about, then you know who you are and you know that you've got that kind of computer. Well, then also you wouldn't be able to use VirtualBox. You'd have to set up a separate Linux machine or just install this software natively on your machine, because there is Windows for ARM machines. All right. Anyways, please in the chat window, go ahead if you have more questions. But something that I wanted to make sure that I emphasized in here, because this is the #1 question that I get on my channel. And let me pull up my virtual machine here. All right, and please let me know if you can't see my virtual machine. I think that this is mirroring properly into the live stream. But, well, let me pull up my channel. I'll pull up my website. I have videos for doing species distribution modeling. And there's one in particular that lets you choose the settings that you'll use to perform the species distribution modeling, and that uses a program called E&M Evaluate. Let me open that up here. Oh wait, bear with me. Here we are. E&M. Now it should find that on the page for me. Hold on, bear with me. It's a little sluggish when I'm running it in emulation. No, we don't need to see that. So what I'm trying to emphasize here, if I can pull this up, well, I'll just have to do this manually, is the way that E&M Evaluate is written has been changed. And so the consequence here is that if you downloaded the E&M Evaluate, the E&M Eval, it's called, package add-on for R a long time ago, or if you downloaded my virtual machine a long time ago, like over a year ago, then there's a different R script that you have to use to run E&M Evaluate. So I know that was a mouthful, so I'll repeat that. If you are trying to use my tutorial about how to get the right settings for performing species distribution modeling, you're going to be running RStudio, and you're going to be running this R package called E&M Eval. Now, the syntax for the people that wrote the E&M Eval package, which is not me, they changed the syntax. So they changed the language that you have to use to run that tutorial. So I actually have 2 versions of that script. If I can just pull it up here, I can show you, that are available. One version of that script is if you are running the old version of the enm eval package, and that would be if you downloaded my virtual machine a long time ago, or if you're just using this tutorial natively on your own computer and you have R&R Studio, and if you downloaded the package enm eval a long time ago, you'd use an earlier version of the script. Whereas see if I can get it here. Whereas if you're using, here it is. Now that's how to install it. I believe this is it. How to decide which settings to use when running MaxEd as well as how to make a bias file. So if we click on Files Needed for Tutorial, instead of just downloading the files that you need to run the tutorial, no, I have it written here. The syntax of the enm eval, specifically of the enm evaluate function, has changed. So if you have an old version of the package, then you would download this version of the tutorial files. If you have the newer package, which is going to be most of you, then you would download this version of the files. Most of the questions I get from my channel, I get questions about all kinds of things, but the number one question is where people are trying to run the script, and then the error message that I see that they're getting suggests to me that they're running the older version of the script here that they downloaded from me a long time ago. So they're probably just continuing to use it, and they've updated the package, or they've updated RStudio and they don't remember it, or they just had my old script lying around and they pulled it off an old hard drive or something. So I noticed that people are using this old version of the script with the new version of the enm eval package, and so I can and the warning messages are very specific. It says something about an error with the tune args, tune.args, the tune arguments. If that's the case, there's a mismatch between the enm eval package that you have and the tutorial files that you're using. So one thing that you can do is come back here and download the other version of the tutorial files and then try running it again and see if that resolves the issue. You can also re-download my virtual machine if you're using my pre-configured virtual machine, and you can therefore have the newest version of the enm eval package, because my virtual machine I update it all the time, and so then you'd be using the new version of the tutorial files, and that you'd be using the new version of the script. So be aware of that. Let's see, who do I have on the live stream here? Let me have a look. Oh, so I've got 3 folks on. Does anyone have any questions for me? Otherwise, I can go on with. I can look up other questions that I've seen on my, that people have left as comments, and I can just kind of go through some of those. This is very much how, for me, real office hours go. Like if this was a class and we were getting ready for an exam, if this was like a study session or something, just try to offer things that would be useful to you. Okay, Dan, I see you've got a troubleshooting question. Go ahead. And while we're waiting on Dan, I will start looking through some old questions I've gotten on my channel. So let's see here. Let's see, what else can I tell you about? I've been moving more and more to, as I was telling you, to using R and RStudio. When I have my tutorials for geographic information systems, I've been doing those using primarily the program GrassGIS. And there's a whole long storied history to GrassGIS. It's one of the oldest GIS programs. Notably, I don't use ArcGIS, which is the one that in your own careers or in your own studies may have been the one that was introduced to you or that is available on your computers. ArcGIS is a commercial program by the company Esri. The licenses, the software licenses are not too expensive, but it's a pain to install, and it's limiting. It means that, for instance, if I have students that are, working with me in my lab, and they're going to be, working on the project whenever they have their time. It means that if we were going to use ArcGIS, I'd have to pay for a separate license for ArcGIS for every machine. It means when I teach my courses and I do these demos, it would mean that I would have to purchase a license of ArcGIS for every one of my students. Now, some universities have site licenses for ArcGIS, which obviates this problem, where everybody who has an email address from that university can download and use ArcGIS, but we don't have one at my institution. And also it limits the reach of these tutorials. So Dan, I see your questions there, and we can work through them. If I make a tutorial for ArcGIS, only people that have ArcGIS will be able to utilize that approach. But if I can demo how to do that approach using open source software, and all of the software that I use in all of my tutorials and in my pre-configured virtual machine is open source, meaning it's all freely available, free to download, free to use, then that means that there's a much wider group of people that can benefit from that tutorial. And in my own life, I almost never use ArcGIS if I can avoid it. There's some very specialized applications where I have to use it, where I haven't figured out the way to do that using open source alternative methods. But otherwise, in my own daily research and in my teaching, I use all the open source stuff. So I actually do use GrassGIS for my research. I actually do use QGIS for my research when I do landscape ecology. Let's see here. So Dan would like to know, Dan is running a SEMOVA, which is a spatial analysis of molecular variants on a data set. And he keeps running into the issue that the bisector is not found. So it will work fine on subsets of the data, and he's unable to find anything regarding the software's limitations. But when you run the full data set, you're running it into an issue. So then it sounds, Dan, like it may be a size limitation. I don't know if this is a particularly big file. So one thing that we could do to kind of get the ball rolling to troubleshoot here would be, bear with me. If you could send me your data files, I set up an email address where I'll know that they're coming from this live stream. youtube@joshbanta.com, as you see on your screen, hopefully. You send me that data file if you're comfortable sharing it, then we could all troubleshoot it together. We can also just go ahead and try using SEMOVA and seeing how it works. I actually haven't used it in quite a while. I know that it's based upon the Harlequin file format. So Samova is one of those programs. Great, yeah, so Dan's going to send that file over, and so that'll be fun. We can play around with that file. This is going to be a big no-no, which is live demoing. You're never supposed to live demo software. That's why I have tutorials that are pre-recorded that you can watch on your own time. Live demoing, things will always go wrong, but that's okay. You know, we're all here to figure this all out together, so it's okay if it's messy and if we're looking at software fail and do weird things. The struggle is the learning. So, a spatial analysis of molecular variants. This is where you're using your genetic data about your population or populations of individuals, and you want to understand what are the natural genetic groupings of those individuals. So if I give you a bunch of DNA sequences of people from throughout the world, and you know, I don't tell you, you know, where in the world those people are from. A structural analysis of molecular variants would allow you to infer, oh, these individuals belong to the same population, which is different from this other population. It's a clustering algorithm based upon the similarity of the DNA sequences of the individuals that you give to it. And so this is really useful, for instance, in a conservation biology context. If you want to know how many populations do I have? Do I just have one giant population that I need to worry about conserving? Or do I have multiple locally adapted populations that are different from one another? So performing a spatial analysis of molecular variants can be a really important troubleshooting, not troubleshooting step, can be a really important initial step in that process. Now, the other thing about a spatial analysis of molecular variants that separates it from other clustering approaches, for instance, you may have used or heard of the approach called structure, called by the name of the software structure, but it's a Bayesian clustering algorithm. The thing that makes a spatial analysis of molecular variants a different kind of clustering algorithm is that it also uses the geographic coordinates of your specimens. So let's say we're talking about crayfish. That's one of the projects I have going on now. And let's say you want to know how many populations of this species do I have. Well, not only does the software look at the genetic similarity to tell you how many distinct groups of individuals you have, but it also uses the geographic coordinates. So it looks for large distances separating different groups of individuals. And then the software is sophisticated enough to see, well, these individuals are genetically most similar to one another, but then there's this other guy over here who's really far away but not close to any other populations. What do we do with that guy? Well, the software can see, well, actually, that individual over there is most genetically similar to these individuals over here, and they're still pretty close to one another, so we can count them all as the same population. So it uses geographic proximity as one of the ingredients to figure out which individuals belong to which populations. So it uses more than just genetic similarity, but also geographic proximity. So that if there's some barrier that's keeping two groups of individuals far away from each other, a spatial analysis of molecular variants can essentially take that into account by seeing, quote unquote, the large geographic distance among those two groups of individuals to further aid in assigning those individuals to different populations. So there's a genetic element and a geographic element. And Samova is a nice introduction to my research and to this channel, because there's both in this channel landscape ecology type stuff, you know, species distribution modeling type of stuff. So I get one audience from that world. But then I also do evolutionary genetics type of stuff, phylogenetics, you know, how are individuals related to one another, and genetic clustering. And those can seem like very disparate approaches that are answering very different kinds of research questions. You know, often evolutionary biology research programs and ecology research programs are very different from one another, but fundamentally they're connected, because the ecology of organisms, their geography, their landscape ecology influences their genetics, and vice versa. So the gene flow among populations is going to be influenced by the topography of the landscape. It's going to be influenced by the environmental tolerances of those species. Are the different populations able to reach each other? So that's not just evolutionary biology, population genetics questions. That's not just about what does the DNA look like and the patterns of gene flow. That also has to do with the ecology of the species and the natural landscape in which those species exist. Okay, so now in real time, I'm going to try to import this file into well, let's see, Dan, I'm not seeing your file yet. Check my spam filter. Otherwise, I can just go through a general work-through of how to run Samova. You've probably already done my tutorial and gotten it to work on the tutorial data. But let's see here. Oh, here it is. Yes, it went in my spam filter. Okay, so I've got your files. So now I've just got to get them. And let's see if I've got copy and paste set up appropriately on my virtual machine. There's a 50-50 shot. So call this Samova. Oh, I'll actually have to download Samova too. Let's see, first of all, can I paste it? No, it doesn't look like I can. Oh, that's inconvenient. Bear with me. Let's see if I can. I can set up that. Let's see, maybe I can drag and drop. Let's see if I've got drag and drop set up appropriately. Drag and drop operation from host to guest failed. See, this is why you shouldn't live demo. Let me think about how I'm going to move this here onto the virtual machine. Because I think I have to update the guest edition stuff. Let's see, drag and drop. Because yeah, it thinks it's bidirectional, but it's actually not working. Do I have a shared folder set up? Okay, bear with me, folks. I'm going to stop the share here, and I'll email it to myself in my virtual machine. Hold on a second, folks. Okay, all right, there we go. Oh, Philippe, I recognize you. So you're from Brazil and you're starting with Maxent modeling? Or maybe actually you could be a different Philippe. But anyway, I'm glad to hear that the tutorials have helped you a lot. That's really great. Thank you for the feedback. All right, so now just hold on while I'm not sharing my screen, I'm going to sneak into my email on the guest operating system and get Dan's files on here for us. Let's look at, while this is loading, I'll look at how many people I've got in the live stream. Okay, so I've got 3 of you in there. It's you know, it's not a huge channel. I think you need 1,000 subscribers to do live streams without restrictions at least, and I think I'm at about 1,500 subscribers. It's been, you know, really terrific to watch it take off, but still a pretty small channel. Okay, let's see here. Sign into my Gmail. Next time I'm going to have the drag and drop updated for my VirtualBox machine so that I won't have to do this kludgy workaround. Any other questions while I'm working on this? Yeah, it's this dead air. But once we get going here, then we can start working through this Samova. Okay, so I've got Gmail pulled up on the guest operating system. I'll pull it up on my machine. Okay, let's see, where is the spam folder? I'll just re-forward it to myself. Sorry, folks, it's for those of you that are just joining, I'm going to be trying a live demo with someone who's having trouble with Samova, but I'm having trouble this time getting it onto my virtual machine. So I am just doing a couple little workarounds so that I can have that up and running for you. Okay. All right, 4 attachments. I'm almost there, folks. Okay, so I got the attachments. Eureka. Now I can share my screen again. Okay, all right. So now let's move this stuff around. So I've got Dan's files right here. I will drag those to the Samova folder on my desktop. And I think I already have Samova installed on this version of the virtual machine. If not, I'm going to have to go grab it. But let's see. I might have to go, you know what, I may have to go open up an older virtual machine. That's another point that I should make, is that I've now split my pre-configured virtual machine into 2 separate virtual machines. I have one for the landscape ecology, the GIS, the species distribution modeling stuff, and I have one for the evolutionary genetics stuff. The reason for that is the evolutionary genetics tutorials tend to use older software that I have to do a lot of tweaking of the Linux environment to get those programs to work right. Like I have to run older versions of Python or things like that, or different versions of Java. It's weird things to get those programs to work, and those interfere with often interfere with GrassGIS or with RStudio, which I use a lot more intensively for the landscape ecology stuff. So I found that it's easier to just have two separate virtual machines, and you'll see that now when you come to my, let's see, do I have Wine installed on here? I don't even think I've got Wine installed in here. Anyway, I should be able to just install Wine on here, and then I should be able to go download the tutorial files and run Samova. So we should be okay. But just do make a note of that. In the past, if you've downloaded my virtual machine, there's a link. It says files needed for the tutorial. If I can find it here, that has changed now. Where my pre-configured virtual machine, it brings you to a second web page where it says, warning. You have to choose which pre-configured virtual machine you want. Do you want the one for landscape ecology, GIS, and species distribution modeling, or do you want the one for population, evolutionary genetics, quantitative genetics, phylogenetics? You have to pick one of those two virtual machines now to download. And of course, you can download them both. But then, depending upon which of my tutorials you're following, you'd use one of those virtual machines versus the other. All right, okay, so let me just go ahead and download the Samova tutorial files on here. So if we come up here to the top, I could be doing a Control F search, but it's not working the way I would expect it because I'm using two different screens right now for this live stream. So I have to just look by eye. Here it is, how to do a spatial analysis of molecular variants. And then, files needed for tutorial. Okay, let's download it. Well, that could take a little while. Shouldn't. It's not a big file, but maybe I just have too many programs running at once. Okay, there it is. Good. Now, I'm going to need to go here in 10 minutes. So, Dan, what I'm thinking is that I'm going to play around and try to run it, see how it looks. And then something else, if you wouldn't mind, can you send me the predecessor files? So presumably you started with these files in, I guess, is it FileML format or FASTA format? Presumably you started with your genetic data in one of those formats, and then you converted it with my tutorial into the Harlequin format. So if you wouldn't mind, could you also send me FASTA? Great. If you could also send me the original FASTA files. And so I don't think I'll have time to do that today, but the next time I do my office hours, next week, my plan is to do these at the same time every week. Hello, Matab, and please forgive me for not knowing how to pronounce your name correctly. And I see you asking, is there a way to contact me through email? Well, there's a lot of ways, but this is a good one here. I have this set up for this live stream for people like Dan, who's sending me some files to tinker with. YouTube@joshbanta.com. And when I do my live stream again next week, I can, Matab, I can potentially, if you send me your files, we could potentially work with your files in next week's office hours. So let's see here. So now, let me open up the Samova tutorial stuff that I got. Yeah, so the virtual machine I'm using right now is the one that's set up for landscape ecology stuff, so it doesn't have the population genetics programs on it. But that's okay. Samova is easy to run. So that there, and then I'll just go here to my desktop. Drag these files out, uncompress them, as you've probably heard me say many times in those tutorials. Yes, you're quite welcome, Dan. That's what office hours are for. The messiness of real problems. Okay. So now I believe I do wine. Oh no, I got to go to the right directory. So let's, what directory am I in? I got to go to CD, desktop. I got to go to CD, Samova. And then I think I type wine, Samova. Okay, looks like wine is running. Okay, so I may need to be better prepared. I see, I shouldn't have installed wine, I should have installed wine too. I may need to be more prepared next time. Yeah, I'll have to look up how to install wine on my machine and install it. Oh no, and please, nothing to apologize for about blindsiding me. I was hoping that people would bring their own data. That's great. This being the first live stream, I just don't have all of my software ready to go. What I need to do is I need to have wine on here. I need to have both of the virtual machines available. So if I get population genetics questions, I can run them on one. If I've got GIS landscape ecology questions, I can run them on the other. But anyway, the advantage is that if you can come back next week, or if you can watch, these videos will be posted after they've been live. They'll be posted for you to watch later. So if you stay tuned a week from now, excuse me, I'll tinker with your data set and see what we can do, and then you can see the outcome. All right, well, I think I'm just about ready to wrap up for today. Is there anything else? Anything else we can chat about before I hit the road here? Let's see, how many people do we have on the... Let's see, how many people have we got on here? Oh, we still got 3 of you on here. Well, let's see here. We'll back up the live stream. Well, anyway, it was great to meet all of you. Again, see the email address on the screen. Great, thank you, Dan. I'll look for those FASTA files, and I'll be sure to set it up so that these don't go to my spam folder. So that email address you see on your screen, that's how you can send your files that you're having trouble with to me. If you send them ahead of time, I can get a chance to tinker with them before our next office hours. Oh yes, you're very welcome, user Bird Drive. Great to see you. Very nice to meet you. Then if there's time, we can work through the problems that you're having in the next office hours. And if you send it to me, well ahead of time, then potentially I could have a solution worked out for you when we meet up again. I can't promise I'll work on everything if this really takes off and I get a lot of viewers, but certainly I'm happy to spend this time just tinkering with other people's stuff and trying to see if we can get it working together. And also, if any of you have any insights when I'm the next one of these, when I'm working through one of these problems, if you have any insight, hey, you could try this or that, then it would be great if we get a community going where people can chime in and offer that kind of crowdsourced help as well. Okay, well, thank you all for joining in on my first ever live stream. This has been very exciting, and I will see you again at this time in a week. So take care, everybody, and have a great week.